We have made a complete spatial and kinetic model of an entire cell over its entire cell cycle.
So JCVI Synth3A is a beautiful organism. This is a little bacterium that is synthetically made, and they essentially cut out every single gene that cell did not need to live and grow. So it went down from almost 1,000 genes down to only 493 genes.
So we were studying this minimal bacterial organism in which the genome has been reduced to half of its size, making it much easier to handle computationally.
We're simulating one cell from the time it's born to the time it divides into 2 cells, which is 2 biological hours.
We have every subcellular network in these simulations. So we have metabolism in there, we have genetic information processing in there, we have growth, and we have cell division. It is a unique simulation.
We were able to do it for the first time in 3 dimensions. We have every single component of the cell in these simulations all at once, the equivalent of thousands of experiments simulated simultaneously. We know the counts of each protein, each RNA, each small molecule, so the sugars. We know all of these things all simulated simultaneously.
You see in these animations the cell starting off, and then it's going to grow. Now you're beginning to see the DNA replication. You'll see more of the large molecules are being made. This is all going on, and that's the difficulty with trying to make these movies. What do you show? If you show everything, it's just a fog. So you have to leave out viewing other things. They're in the simulation, they're just not in the visualization.
And so we break down the space of the cell into individual little cubes, and then each little cube gets simulated by one individual little processor on the GPU. And being able to simulate all of these things in parallel is why we can do this now. It takes us 1 week to simulate on a computer. If you were to try to simulate them all one after the other, it wouldn't happen in a human lifetime.
So what's the real benefit of these 4D simulations? So every biomolecule is in there in its position in terms of X, Y, and Z as a function of time. And if you want to study, say, for the creation of the ribosomes, you can just pull that out and visually look at that in 3D, or let's look at that also coupled together with DNA replication. That's the cool thing with these simulations. Every biomolecule can be observed that we have in the simulation, and it can be plotted out.
These models will certainly open up new research avenues.
Something new will come up. Science does not sit still. I'm more excited about science now than ever.
What would you do if you could ask, I can measure every single thing in the cell all at once? What do we want to measure? The possibilities are almost endless. From changing what the nutrients are around the cell, what it's growing in, to changing how strongly a gene is expressed, to adding a new molecule, maybe a drug. All of these questions are now fully open doors that we can walk through. And the question is, who's going to ask what first?